we're anxious to hear about the bugs and what the bugs ask, so we will get started. It's really great to welcome to our spring Fellows Colloquium. It's a great time to get people together, and as I look out here, you know, we got lots of students here, we got faculty, and we got staff, and it's and friends and fellows, and you know, it's a really nice mix of group of people that we always have at these colloquia. So welcome to everybody. It's always nice to have these kinds of events mixed in with all of the other things that are going on in our lives. So just want to welcome a couple of our fellows and recognize them that are in attendance today. Bob Mikazaki, right here, who's on the alumni board too. And then Don Reich, who's also one of our fellows, who's here. So Don's right there. One of the things that I always like to point out is that we here on the beach, everything that we do is all about our students. I mean, we all have our jobs and what we're doing, but in reality, what it comes down to is our students. It's the university for our students to come here and learn and find out things that will help them for the rest of their lives and careers that they want to go into. And so as faculty and staff and administrators, we're always looking at things that can really support them. So it's always really great when we have students who come here. We have a whole table back here of current students of Dessie, and we have a couple over here that were former students of Dessie. So welcome. It's nice to see you here. One of the things also that I look at is our faculty, and they do a fantastic job of teaching and supporting students in research. And that's what we try to do at the Colloquium, is really point out the kinds of things our faculty are doing. They are doing things that are truly outstanding. Their publications are always with students. They're out there giving presentations. What they do in their research programs with students is just really, I think, phenomenal. I look at the kinds of things they're doing. I just want to point out some of the things then that's happening in the way of recognizing some of these people in our college. Some of us started the day really early downtown at the Hyatt with it was the Long Beach Chamber, but also the Euclid Center for Ethical Leadership, which is here on campus, was recognizing someone for their ethical leadership, and that was Dr. Chris Lowe, who's one of our faculty members and in the Department of Biological Sciences. I know many of you know Chris. He's out there with his students doing all kinds of research out with sharks and whatever, but he gave a really great presentation about how we always think sharks are, and a lot of our sea life out there with everything that's happening, global warming, all these sorts of things, are disappearing. But what he actually showed is there are a lot of things that have been put in place in the way of like the Marine Mammal Act, getting rid of gill net fishing, those kinds of things that have really made a change, and a lot of these things animals are coming back. You look at the increase in various kinds of fish, you look at sharks, you look at marine mammals, you know, the numbers are actually on the incline. So what he was saying is that there are a lot of good things that we are doing out there. One of the things I think Chris is so great about doing is he always promotes his students, and that's what he did this morning. He said, it's like, I'm only, you know, partially responsible for all the research we do, it's his students, and I think that's what all our faculty do, and that's why Dessie students are here. You know, it's about our students and what they're learning. In about a month or so, we're going to have the University Awards celebration, and we always have some from our college who get awards at these celebrations, but I just really proud. This year we kind of did a sweep almost of people who are going to be honored. Diane Graham, who's our technician for all the microbiology program, is the outstanding staff member. Steve Mesick, who's in the chemistry department, is getting the Provost Impact Accomplishment of the Year in Research, Scholarly, and Creative Activities Award. I think that's his second time. Brittany Dawes is one of his students, gets the Outstanding Undergraduate Research Student Award. Min Min Ho is one of Deborah Fraser's students who's in biology. She's getting the Outstanding Graduate Research Student. Kaylee Fruman, who's an advisor and immunologist in the Department of Biological Sciences, is getting the Distinguished Faculty Advising Award. Ted Stankovich, who's a faculty member in biological sciences, getting the early. Ted, you're right here. You are. I did see you. Congratulations. He's getting the Early Academic Career Excellence Award. It's just congratulations. That's really cool. And then Chris Lowe's getting another award, the Distinguished Faculty Scholarly Creative Achievement Award. So I'm just, I'm not amazed because our faculty and our staff are so good. It's just nice to really see them being recognized for all the things they do. And I hope our students actually appreciate that too, that they have a lot of great people to work with here. So tonight we're showcasing a faculty member from biological sciences, but she's also an interim associate dean for research in the college for this year, and we'll see where things go. We have a search going on, so we'll see what happens there. But I'm going to turn it over to Brian Livingston, who's the chair of biological sciences, to actually introduce Dessie.
All right, thank you, Laura, and thank all of you for coming. It's my pleasure to introduce Dessie today. It's very important that we educate our students about the changing environment. We live in a very complex urban environment where we interface with nature, especially the ocean, and one of the critical components that we have to deal with is water, all right, either the lack thereof or when it comes pouring down and washes everything out into the ocean. And then when we go surfing, we get eye infections, Bob. So, yes. So I think it's critical that we educate our students about this, and Dessie does more than that. She's at the forefront of not just telling our students about the environment and the changes and how that impacts human society, but she actually trains them how to assess that, how to look at the changes and see how that affects the organisms that live here. So I think that's something that's really important that we should continue in our department. Dessie basically bailed out of high school and went and got her degree in biology from California State University of Fresno. She then got a PhD at University of California, Davis in ecology. And she came to Cal State University Long Beach as an assistant professor in 1999. And she started a program in entomology, studying the madrone butterfly, which lives in Mexico, right? And had a research program studying that butterfly until she actually developed an allergy to that particular organism, which put a stop to that research program. But she transitioned to the program where she started studying assessment, all right, of the local environment using the insects that she was trained in, all right? And actually now what we'll see now is that she's actually expanded upon that, is looking into water using other organisms and looking into bioassessment as a much more expanded capability of sort of assessing how our environment is changing and how we can fix that. All right? So I think that what Dessie has exhibited is that she's faced changing environments and has been able to adapt and be successful. And we'll see if she can tell us about how these organisms are. She actually served as my associate chair for many years in the Department of Biological Sciences. When I took a sabbatical, she came on board and took over as chair. And now she's moved into the college and is working as associate dean for research. So I think that she's really contributed in many ways, and I'd like to thank her, and we'll look forward to hearing what she has to say. So, Dessie.
Thank you, Brian. That was great. Thank you all for showing up. Okay, so I had this talk. I'm going to try to make it not too crazy technical, but I think you guys are going to enjoy it. So we're going to just jump in. So, ask the bugs. Water quality. So if you've been paying attention to the news, you know that Flint, Michigan has a problem. And they don't know whether the water in their glass is something they should be drinking. And of course, they know what chemicals are in there that are bad, lead, right? If you go out to a natural system where there's a stream flowing, it looks good, doesn't it? That water looks as good as that water, but is it really healthy? Is this system really something that that's going to be sustainable for the long run? And so what you would need to do is we have to assess the quality of that water, and typically, traditionally, you would go out and take a sample of water and you do a variety of chemical analyses. So as long as you know which chemicals to test for, That's the way to do it. The problem is that in nature and in naturally occurring water systems, there's all kinds of chemicals, and the chemicals interact with one another in weird ways sometimes. And so chemical analyses there's a limit to how useful they can be in telling you how the quality of water is. More recently now, and this is actually, California's been behind the curve. New Zealand and Europe have been doing what's called bioassessment for decades before us, and California just started doing it in 1998, 1999. And so, but originally it started using fish in the Pacific Northwest, and unfortunately we can't use fish in California, Southern California, because a lot of our streams are naturally fish-less. There's not fish there. The fish that we do have were put there by the Department of Fish and Wildlife. And so what we do is we have another assemblage of organisms that we can use. And so this is one of my favorite little bugs. Isn't it cute? Look. So this is the top of it. So normally it runs around on the rocks like this. This is its undersurface. And these are all these little suction cups. And so it sits on the rock, and it's like a little aquatic cow. So it eats all the periphyton, which is very often they're what are called diatoms. So there's the little cow, and there's the grass. And so the diatoms, which are part of the periphyton, are the primary producers in these systems. And then you can have the insects, which of course eat that. And then of course there's other insects that eat these bugs. And then the fish comes along and eats the bugs that ate the other bugs. So what I'm looking at are the very bottom layers of these food webs. Dr. Livingston mentioned that we were at or I guess it was Laura, this morning at Chris Lowe's talk, and he's at the other end. Sharks, the apex carrot. Right now we're going all the way down to the bottom now. Now we're at the little tiny things, Okay, so looking at the biotic measure or a bioindicator can add a lot of value to our assessment of a given system. And that's because a lot of these little creatures that live in the water are long-lived in the water. So some of them take several years to mature. Most of them mature and become adults that fly around. So they not only are in the water, but when they come out, they interact with a riparian habitat as adults. And then they're food for other things that people care about. Most people don't care about the bugs. They care about the bugs as food for other things. So fish or birds. So anyways, it can give you an idea of the health of that system. And because they're in this system for so long, they're indicators of not only current conditions, but recent past impacts. So if something happened 2 years ago, and you expect to see a certain taxon there, or a certain type of species there, and it's not there, you have some inkling that something happened in the past. If you go out and take a water sample, all you're going to get is right now, here and now. And so doing the chemicals doesn't allow you to have this sort of temporal sensitivity. And upstream stuff that happens flows downstream. And so you can have impacts on a downstream community that give a sort of a holistic view of the health of the entire system. So that's definitely added value. Benthic macroinvertebrates, BMIs, as bioindicators are especially helpful because some of them are very sensitive to specific impacts. So some of them are very sensitive to sedimentation or water, the temperature of the water. Certain kinds of heavy metals will cause weird things to happen in in specific bugs. And so the presence or absence of the organisms can give you indications of certain kinds of conditions. And then the complexity of the community, because it's not just little aquatic cows walking around. There's things that eat those, and there's other things that eat them. And so if you have trophic relationships that are really stable, it indicates that this system has been temporally very stable for a very long time, long enough to build up those populations of primary consumers, the little cow, and then you can also get the little aquatic cougars and things. So, so in order to get that, you have to have a long temporal stability in the system. And then we're always interested in change, and if you see a change in the community over time, it implies that there's a change happening. And so that way it's an early indicator then of that we need to find out what's going on. And then furthermore, you can't actually measure every possible stressor, but you might want to know, is this system healthy or not? So if we knew that lead was a problem, you go out there and measure lead, but you don't know what's the problem. And so being able to look at the whole community, it actually acts as a first alarm when things are declining or when things are getting better. So, like I said, this is relatively new in California, and in Southern California in 2005, Pete Odie, who's part of the Department of Fish and Wildlife, published a paper, he and two of his buddies up there. And it's based on a number of sites in Southern California. We have two different ecoregions down here. We have the mountains and we have the basically the arid areas. And they screened a whole bunch of different candidate metrics to try to understand, if we take a community of bugs, can we boil it all down to one number that gives you some indication of health? And they call that an index of biotic integrity, or an IBI. They identified 7 metrics. I have a very complicated slide at the very end. If you're interested, I can share it with you, but I'm not going to share it with you now, so just trust me. Okay, and so here's Southern California. There's the darkened spot, is this big map here. And so we have these 2 different ecoregions, and these are all the different places they sampled. Notice there's a big blank in here. So there are problems with this index because not all of our types of streams that we have, say, in this area are part of the model experience. So they built a model based on these sites, but if the sites weren't included, then sometimes you have issues in how applicable that model is if you're outside the model experience. The IBI has now been replaced recently with what's called the California Condition Stream Index, the CSCI, and we're taking all of our data and converting it now into the CSCI from the IBI. In fact, Lauren Fay, one of my grads, just finished her master's last December. She's on the GIS now, the Geographic Information Software System, to delineate watersheds and recalculate all these indices. Okay, so who are the BMIs? I'll just give you a few pictures here. So true flies. So these things are all aquatic as nymphs, as young things. Here's a stonefly. Here's a caddisfly. All that little fluffy stuff, those are gills. So they're pretty cool. Here's a dragonfly, the nymph of a dragonfly, and that's a Femeroptera, mayfly. So these are all some of the big things that go into calculating these IBI scores. Okay, so we got our first contract from the State Water Resources Control Board in 2005. And between 2006, we started sampling, and between 2006 and 2011, we sampled 182 sites in Southern California. The state is actually divided up into 9 regional water boards, and Region 8 is the one we have, which is the Santa Ana River. So here's the Santa Ana River and all of its tributaries, as well as the San Jacinto River, which I don't have it on this map. And here are the IBI scores, and so this is just like A, B, C, D, E, F. No E. So A, B, C, D, F. So if you have a red dot, you have an IBI score between 0 and 20. Actually, it should be 19. Past student Ian actually did this. I just noticed that today. I was like, oh, that's supposed to be 19. Well, and then so if we started the A's, if 80 to 100, that's an A. 60 to 80 is a B. That's a C, and that's a D, and that's an F. Okay, so it looks bad, huh? It's not surprising though, right? I mean, you think about what these streams are, and some of these streams are not actually naturally flowing streams. The only reason they're flowing when we go out and sample is because of urban runoff. And if the people weren't here, there wouldn't be any water to collect the bugs, and there wouldn't be a red spot there because there wouldn't have been any water there. So it's kind of a funny thing, right? But anyways, we have water there, and we're charged with monitoring or keeping track of how good or how bad the water is. Obviously, the upper elevation sites score better, but not all of them do. I'll let you look at that in a minute. Yeah, what happened to Fish Creek? Yeah, you know where I'm going with that. Okay, all right. So, when I first looked at this, I thought, oh my gosh, this is so sad. We should just all go shoot ourselves or something. You know, there's no point. And then what I did instead was I said, okay, well let me divide this big region up into biologically relevant subregions. And I did that based on the bugs and also its physical habitat characteristics. So when we go out and collect the bugs, we also characterize the stream, what kind of substrate it has. So is it boulders or muck? We collect water for chemical analyses. We characterize the riparian habitat. There's a 23-page data sheet that the crew has to fill out every time we go collect bugs and water. And so I put this in. I used multivariate statistics and figured out which ones were. And I divided up this region then into 4 subregions. So basically, it's low elevation Santa Ana, mid elevation, and then upper Santa Ana, and then the San Jacinto region is on its own. And then I thought, okay, that's good because it's not they are all equally bad. So let's compare apples with apples. And so I took within a subregion, I got the mean of all the IBI scores and a standard deviation around that mean, and I said, okay, do I have sites that are around, that are the average as well as within one standard deviation of the mean, and do I have sites that are below one standard deviation of that mean? In other words, they're really bad relative to everybody else in that population, and are there sites that are one standard deviation above the mean? In other words, these are the good ones. And then I plotted that. Okay, so this is the Santa Ana River. Here's Prado Dam right here. And you can see that there's some sites along the Santa Ana which are much better than other sites. Because we have variation in these data now, we can say, okay, well, why is that? What's going on there? And then, of course, we have these ones here which are one standard deviation below the mean for this particular sub-area. And I think, so what we're doing with this information, it goes to the State Water Resources Control Board, and now we're doing what are called causal analysis or causal assessments to try to figure out, well, what's going on there? And how come this one is so much better than that one? It's right downstream. And so this allows us to have a window into what's going on biologically in these freshwater systems. Here's another one of these sorts of maps. Here's Big Bear Lake. Here's Fish Creek over here. So now, these numbers, by the way, are the actual IBI scores. I should have mentioned that. So we have, there's a 1. That's 1 out of 100. That's 33. Okay, that's not, all right. And then we go higher elevation, and now we actually have sites that are 60, 77. So those are actually Bs. We had, I think, one site that got an A. After all those 182 sites, we got one that had an A. But this allows you to say, okay, well, what's going on here? What is happening in this particular site? Why is it so bad? And what's happening in these other ones? And why are they so good? And then, of course, so this allows us to figure out, to start asking those questions and getting answers. Another thing that we did is, so we had those 182 from 2006 to 2011, 182 sites. And then I had all those that I labeled them. Is it a red site? Is it a green site? Is it a yellow site? And then we went back to all the red sites and all the green sites, all the really bad ones and all the really good ones, and we resampled them. So these are the ones we resampled in 2012. And the next slide I'm going to show you the ones we resampled in 2013. And now what I have here is that if it's a red dot, that means the IBI score actually went down by 5, at least 5. If it's a green dot, it actually increased. In other words, the quality of the habitat or the bugs that are there actually got better by 5 points. And then yellow is it was between plus or minus 5. So now you can get an idea for the region what's changing over time. And here's the Santa Ana and Prado Dam, and this is terrible, minus 16, minus 24. And this one, however, this is Timoteo Awash, actually got better. And if you go to the next, so that was 12. And this is 13. So you can see there's still a lot of, so it was consistent because we couldn't sample all of them in 1 year, so we had to break it up over 2 years. And then you can see this one again is looking, this particular watershed or waterway right here is looking pretty good. I think what happened here is they started adding more water. They started releasing water out of, so that's Beaumont right there. I think there's a water treatment plant over there, and they started releasing more water. Because when we went there, we was, oh, there's more water here. When it comes down to it, it's all about how much water is there, really. And then this is weird. Okay, so this is Idlewild. This is Strawberry Creek. And it's red. And then these are changes. Actually, let me go back. Oh, I don't have that one. Oh, yeah, I do. Then go back. No, I don't. Okay. Strawberry Creek is this little creek that flows through Idlewild. It's a high elevation site. And so we have minus 10, minus 14, and plus 37. And then the next year, minus 53 and plus 29. And there's a town there, and there's something going on in that town. And when we go sample there, we have to, knock on doors and say, hey, we're going to sample some water. The water, it's God's water, right? So they have to let you on their property so you can get to their water. Sometimes that doesn't work. But anyways, there was a guy there that actually said that it's God's water. It's okay. But there's uranium, naturally occurring uranium up there. And so there, when we were up there, you were looking for that uranium? And I'm like, no, I don't think so. But anyways, that's a study that needs to happen there. What is going on at Strawberry Creek up in Idlewild? Okay, so that's one way to use bioassessment. In 2013, we got another use for bioassessment, and this is a sad thing happened. So here's Big Bear Lake. Cold Creek is a little creek right here, right there, and that's so you can get oriented. And what happened is that in April of 2013, this truck He's driving, big truck driver, full of fuel. So it had diesel fuel and gasoline in his big double tanker thing. He's driving too fast, flipped the truck, and all these gallons and liters of fuel went into that tiny little creek called Cold Creek. This creek flows into the Santa Ana River, which is part of part of our metropolitan water source. It was a huge issue. They jumped on it within 24 hours, and in November we were actually contacted by so this is confusing. So it's in a creek that's in the San Bernardino Mountains. So we have the San Bernardino National Forest. It's a creek. So we have Region 8, the State Water Resources Control Board. There's potentially an endangered species there of birds. We have Department of Fish and Wildlife. Is there one more person? And then we have the insurance company of the truck driving company. So every time we have a meeting, there's like 30 people, it seems like. There's 20 people for this little creek. Anyway, so we got the bioassessment contract in November of 2013. What happened was, whoops, go back here. That's how, can you see the tiny little bit of water? That's the tiny little stream. It's a little stream. It's like this big anyways. So they jumped on this, and they had all these absorbent pads, and they brought in truckloads of water, and probably more damage due to all that than the oil itself. I don't know, maybe not. They spent almost $4 million on the cleanup. And then they contacted us and said, well, is it safe? Is it healthy? Did we do a good job on the cleanup? So we designed a sampling protocol that looks like this. So here's Highway 38. So Big Bear is up there somewhere. The lake is up there. Angeles Oaks is over here. Actually, it's right there. And here's Cold Creek running down here. And here's the Santa Ana River. The Santa Ana River flows like that. So here's the spill site on the road. We sampled upstream, immediately upstream from the spill site. And we sampled immediately downstream. That's where that previous picture was with all the absorbent pads and whatnot. And then there's a little hike. You hike down here and you go out to this other site here. And then you run out of trail. And then you go cross country. Oh, that should be a 3. Typo. Okay, that should be site 3. And then site 3. And site 3 is like you have to go cross country down. It's nuts. I did it twice. Ted did it once. He survived site 3. It's crazy. I don't do it. That's why I have all those young people over there. And then we have another creek that's over here. That's just the next creek over. It's called Forsey Creek. And the idea is that this site up here and this creek should represent what it would be unimpacted by this fuel spill. Granted, ideally, we would have had data on all these sites before the fuel spill. But who knew, right? Okay. And then also the other question was, is this Cold Creek, is this spill flowing into the Santa Ana River having an impact? So we had site samples that we did below the confluence and then above the confluence. And then we made comparisons with this. And we sampled benthic macroinvertebrates and freshwater diatoms. Okay, so here's our biological indicators. So these are some slides actually from Robert Esquivel's thesis, which it's almost done, right? No, he had it almost done, and then he kind of had a little heart attack and disappeared for 3 weeks. I go, Robert, what's going on? He goes, oh, you can do this, and he's going to do it. Yep, he's going to do it. Anyway, all right. Okay, so I talked a little bit about the bugs. So these are some cool pictures of bugs, but I haven't said anything about diatoms. In 2013, I applied for a sabbatical to go to the diatom boot camp. In Iowa, and I was granted that sabbatical, and I brought diatoms back to my lab. Same time we got this contract with Cold Creek. It's crazy. I did the boot camp before we got the biomonitoring contract. So I put the diatoms into the contract, and nobody said anything, and so now we're doing them. And now I have several diatomists. I have Sergio and Brina and Eileen are all people in my lab that are doing diatoms. They make wonderful, beautiful slides. So what are diatoms? I'm going to take a little side trip here. So diatoms are these single-celled organisms that are photosynthetic. And they're very tiny. So if you were to put this one end to end, you'd have to have like 100 of them to get a millimeter. Okay, they're tiny. And the diversity is amazing. So there's a little diatom, that's a diatom. These are individuals. That's an individual. This is a colony. So you can see they're like little, tires all stuck together. Each one of those little things is an individual diatom. Here's also individual diatoms. And then if you break them all up, they look like that. There are over 1,400 freshwater species in California. And there's, I don't know how many species of marine diatoms. And the really cool thing is that they have a shell. It's not a shell. They have silicates in their cell wall, which is glass. So they have a glass cell wall, and how you identify them is you bring them back to the lab and you clear them of all their organic material, and you're left with these frustules, which is basically the shell. And then you can identify them from there. That's why I thought I could do diatoms, because they're basically just tiny little photosynthetic insects, because we identify insects based on their shells. They're not really shells, right? So a little more interesting things about them is that they, up to 40% of the Earth's oxygen is considered probably diatom of origin. So that includes all the marine diatoms, of which there's lots, but also the freshwater ones. They're also potentially going to be used as a biofuel, because some of these are chloroplasts in here, but a lot of this is lipid. They produce their own lipids. And so sometimes you'll go out if you're hiking and you see a stream and there's like this sheen on it, and you're like, oh, there's oil here. How did oil get here? That's what I always thought. But no, it's diatoms. They have this, yeah, it's cool. Okay. Another few little things about diatoms is you maybe know about diatomaceous earth. Okay, well, there's the word diatom in there. It's there for a reason. Diatomaceous earth is our marine sediments, where we had all these marine diatoms. They die, and their little frustules float down to this. Remember, they're tiny, tiny, tiny little things. There must be I don't know. We don't have a number that big for how many diatoms are there. Look, there's a tractor. How many diatoms? Anyways, so what's left over is the little glass shell, and so they're used in all kinds of applications, like for filters. So like this last one, you can see, if you wanted to really finely filter something, you'd run it through that little shell. And you can use it also as a non-toxic way of killing arthropods. So here's a house centipede, and because the diatomaceous earth is all these little pieces of glass, and arthropods have this external skeleton, what happens is that the little pieces of glass stab them, and they open up all these holes, and then the little arthropod desiccates or dries out, and then you have this left over, and it's completely non-toxic. So I was getting these pictures for this talk, and I found out that now people are eating diatomaceous earth. What? Have you heard about it too? I don't know. Anyway, I wouldn't recommend doing that. Anyway, so there you go. That's enough about diatoms. I don't know. Okay, so we collected at those sites. Remember the sites we have? We have site 1, 2, 3, and 4, and Forsey Creek, and then Upper and Lower Santa Ana, and usually do 600. So do you remember, Robert, how many?
It was 28,000 benthic macroinvertebrates and 32,000 diatoms.
Yeah, that's what we've done so far, and that's his thesis is all this Cold Creek stuff. And then we transform it for a reason, so we don't, so dominant taxa are not really influential. We calculate what's called a Bray-Curtis similarity, and I'm going to show you another picture about that. But basically, this similarity index varies from 0 to 1, and what you're doing is you're comparing not only the types of species that are there, but their relative abundances in these two samples. And if they're identical, then you get a Bray-Curtis of 1. If they're completely distinct or completely different, you get a 0. And so this is sort of visualize a Bray-Curtis. Let's see if this works. I've never done this with a crowd before. So think about it as a distance measure. And maybe some of you guys remember we used to get these in our maps, so you'd know how far you had to drive. Now you just get your phone out and you stick it in there, it tells you. With traffic, it'll take you this long. Okay. So this is a distance in miles or kilometers, and you can look on here and say, oh yeah, Eureka is 476 miles from Yosemite, and it's that far from, okay. And if you just had this information, you probably could put together a pretty good representation of how far apart all these things are in two-dimensional space. And that's basically what these programs do for us. Instead of having distances in terms of miles, we have distances in terms of how similar they are with the organisms that are living there. And then you can plot it on a 2D graph. So here's our sites. Remember we had site 1, site 2, site 3. These are immediately downstream from the spill. Site 4 was immediately upstream. And then Forsey Creek was that little creek that was on the next watershed over. And then we had our Santa Ana sites. So if two dots are close to one another, that means their distance is really small relative to how similar they are to other sites. And so this is what it looked like when we first went out there in May 2014. And these are the benthic macroinvertebrates. So what this is telling you is that right immediately downstream from the spill and then that little tiny hike, these are all indistinguishable from one another. So these communities are very similar. Site 3 is different, okay? And the Santa Ana sites, I would say that they're not different from one another, which means that this spill, at least now when we went out in May, is having no appreciable effect on the benthic macroinvertebrates in the Santa Ana River. Yay. But there is a change when you go up, because Site 4 and Site 1 are like, I don't know, 0.2 kilometers from one another. And the only thing that's between them is that road and that spill. Yeah, they could have been this way to begin with. It's probably not likely, though. And the fact that 4 and 4C are coming together means that Site 4 is very different from these other ones. And then this is the fun thing about having two different bioindicators. They're actually sort of, they agree with one another. So these are the diatoms. The diatoms are more distinct. And in fact, it's one of the things that Robert and I were talking about. Their community changes over time after a disturbance are on a different time scale than the bugs. Who knows why? Maybe because they reproduce so fast. Maybe they should get to equilibrium faster then. I don't know. Maybe, well, we're talking about it. So here's Site 1 again. Site 2, Site 2. These are all distinct. But Site 4 and 4C are still together, which means that this really does suggest that there's an impact. Even after they spent all this money on the cleanup, there's still an impact on these communities. And the Santa Ana River, though, is showing no impact at all. So that's good, I guess. Okay. One of the things we also noticed, so here's some of the diatoms from Site 2, I believe. So here's a normal one, and then here's one that's not quite normal. And here's a normal one, and here's one that's kind of all bent. And this one's normal, and this one's called cha. And then here's diatoma. So this is one of the more common ones. This is, so we had some that looked like this and some that looked like that. And so you know what's funny is that when we first started doing this, we started with these sites that had these weird diatoms, and we're like, we can't figure out what they are. What is that? It was like, oh wait, they're messed up versions of that one. So we had teratologies or deformities in the diatoms right after we did that first sampling. And these were more common downstream from the spill site. Now we have 4 different sampling periods, and those teratologies are almost gone. Is that right, you guys? Yeah? Okay. All right, so here we have 4 different, now you can see what the change through time. So here's our original May. Notice May, 1, 2, and 3, the sites are clustering away from the other sites. Here's Site 3, which is downstream from it's the furthest point from the spill. You'd expect this to be the least impacted, at least in the, okay, sorry. So those are the January and May are here. And then June, which was the most recent time we sampled, and we have to go out there again this year. Here's Site 4 and Site 4C, and then of course the Santa Anas. And so the line tells you what degree of similarity. So this gives you statistical inference then on what the impact of this system was. So there's no impact on the Santa Ana at this point. Yet there's still an impact on these other sites because eventually 1 and 4 ought to get together, right? Assuming they were the same to begin with. Those are the diatoms, and then here's what the bugs look like. So again, this was our original sampling immediately, well, it was a year and a half after the spill. And here's Site 3. And then 4 is still up there with 4C, so it sort of agrees with what the bugs were saying about where this system is. And so eventually, ideally, and maybe it will never happen. I mean, this is a big question philosophically. In when you have a disturbance in an ecological system, do you expect it to return to its original state? Or does it just go to a different state? And so we may never see these things come together again. We'll have this contract for biomonitoring forever. Okay. All right, so here's acknowledgments. I want to thank Marianne Horton and Raquel for organizing this great event. The Stream Ecology and Assessment Laboratory, I have some past graduate students who were involved with that data set, and that's Marty, who's my spider guy, and Lauren, who's over here. And then we have current grad students. We have Robert's here, Matt's not here, Eileen is here, and then Jose, and then Fritz. He's not my grad student, but he graduated, and he's doing, what are you doing now? Teaching credential, there you go. So that's sort of a graduate student. And then the undergrads that were most involved is Sergio and Brina, and those are my diatomists. And then there's noon ones. I didn't know you guys were going to come. So I have Brittany and Manit are also. They just joined last semester. Yeah, two semesters. Jesus. Okay. All right. And our funding sources, the Surface Water Ambient Monitoring Program, which says SWAMP, This is where we upload all of our data. This is where we get, the money from SWAMP goes to the state water boards. State water boards gives me money, and then I give them money. So it works out. And then nationwide is, the agribusiness insurance is the one that's funding the Cold Creek Monitoring Project. And that's it. Oh, and Heather Boyd, she's the Santa Ana Regional Water Quality Control Board person.